studies. Apart from that, we are able to see that they maintain blood pressure, the plasma proteins, and the albumin, it maintains the osmotic blood pressure, maintains osmotic pressure of blood, and apart from that we have clotting proteins and antibodies. So the albumin is the most abundant plasma protein, and they are called colloids, which hold water and are responsible for blood colloidal osmotic pressure. That is, it will retain blood within the blood vessels, and if the amount of albumin is less, then more fluid will come out of the capillaries that will fill into the tissues and leading to swelling. So it is responsible for blood colloidal osmotic pressure, that is albumin part. Then again we have even globulins which are present. So they are also of different types like alpha, beta, and gamma, and they are mainly concerned with the transport of different substances like lipids and ions. So like gamma globulins are the proteins, are the protective proteins, basically the antibodies. Lastly, we even have fibrinogen, the clotting proteins which prevent excessive blood loss. So this was all about basically the proteins. Now, talking about erythrocytes or RBCs, red blood cells, they are anucleate and they lack nucleus, and they're filled with basically hemoglobin, which carries oxygen. So these are biconcave discs, which have a great surface area for gas exchange. This biconcave disc, only this shape of this is helping in to exchange gaseous exchange, and the half life, basically we say it's 120 days for RBCs, and they contain 280 million hemoglobin with 4 heme chains that contain iron, containing the iron part. Fine. So in red blood cells, we have 12 to 16 gram of hemoglobin is present in 100 ml of blood. Since RBCs are without nucleus, so they have limited lifespan, that is 120 days. And after this, the RBCs are removed via spleen, right? So also spleen separates old RBCs and dead RBCs, um, right, and are degraded in the spleen and in liver. So if we go to the next slide here, so this slide shows the biconcave shape of the RBCs. So this increases the surface area, so more oxygen can be carried, and there's not having any nucleus as such. So this is the basic benefit of red blood cells, right? So red blood cells basically are primarily formed in the bone marrow, and in prenatal development, hematopoiesis is in the yolk sac and then in the liver and spleen. So, and in case of the newborn, all erythropoiesis that occurs in bone marrow. So in case of formed elements, RBCs and leukocytes. So formed elements, we have red blood cells and leukocytes. So RBCs are basically, we can say, the flattened biconcave discs, and they provide increased surface area for diffusion. So lacking nuclei and mitochondria is a very good part, because that only helps in maximum achieving the maximum gaseous exchange. So each RBC also additionally contains 280 million hemoglobin units, right? So now, in addition to this, if we see, so RBC consists of cell membrane and some proteins associated with the membrane also, and a lot of hemoglobin is present inside it. So if we say in lengthwise, we can say RBC is a 7 to 8 micrometer, and in depth, if we say, and the diameter part, it is approximately 2.5 micrometer. And in the most center point, RBCs are just like 1 micrometer. That is the overall concept of the, you can say, red blood cells. Apart from that, RBCs have extra membrane, and benefit of this is that it's biconcave disc, so that surface area is more, so that hemoglobin can exchange the gases easily. And the extra membrane, it helps in folding of RBC. That is when RBC has to pass through narrow capillaries, and hence they are flexible and fold on themselves. So that is the beneficial point. Otherwise, the RBCs are going to lose that membrane, and finally will quickly result in death of the RBCs. So that's the main problem RBCs have. Now we have this hemoglobin. Discussing about hemoglobin, so if we see, during synthesis of hemoglobin, first molecule which is made is called that the pyrrole ring, and 4 pyrrole rings are assembled, and in the center iron is put, and that finally, and we synthesize, and there is a synthesis of globin chain. So finally, specifically the erythroblasts, they produce this protoporphyrin ring, pyrrole ring. So, and in center there is iron, then heme part, and finally we have this, you can say, globin chain, which is attached to uh, the part we see that hemoglobin formation. So this process of hemoglobin synthesis basically is occurring in a cell, and that is the hemocytoblast. And we have certain reactions also which go on